So Bo-yong, you ready?
Yeah, sure.
Okay, cool. So good afternoon and welcome everyone. My name is Boris Bernhardt, and it's my pleasure to host the Feindel Virtual Brain and Mind Seminar Series today. Before we get started, I would like to ask you all to please mute your microphone and to stop your video so that we don't interfere with the presentation. Thank you very much. This series is co-organized by Nathan Spreng, Bratislav Music, and myself, and we get support from the Neuro events team, and in particular Sasha Kelly, who is also participating in the session. So in the lecture series, we try to feature both early career investigators as well as early established researchers who cover a broad spectrum of methods and research questions present at the BIG. For today's session, it's my pleasure to introduce Dr. Bo-yong Park, who's a postdoc in my lab. Bo-yong carried out undergraduate and PhD studies at Sungkyunkwan University in South Korea, and he worked briefly at the Max Planck Institute of Empirical Aesthetics before joining my lab. Dr. Park has exceptional expertise in multimodal neuroimaging, machine learning, and all kinds of data science questions that we usually apply to analyze connectomes. And today he will talk about the application of manifolds to structural connectome data in the context of the study of typical and atypical development. Thank you, Bo-yong, for agreeing to give the talk. For everybody who's in the audience, please hold on with your questions until the end, and we'll have a formal question and answer period when Bo-yong is finished. All right.
Thank you. Thank you for the nice introduction, Boris. Thank you for having me today. My name is Bo-yong Park, and today I'm going to introduce how the structural gradients can be applied to the typical and atypical developmental conditions. The neuroscience is the scientific study of the nervous system, and it is a highly multidisciplinary field that combines the study of molecular, circuit, and system-level mechanisms using a cutting-edge experimental and analysis techniques. And multiple technical innovations allow increasingly detailed investigation of brain structure and function, and understand the features of the neuroorganization. And at the same time, the increasingly complex data requires the demands for developing novel analytical techniques that can handle such high-dimensional brain data. Here, one of the issues for analyzing the brain signal is how to represent such a high-dimensional neuroimaging data. So we can first simply think about a method of compressing such high-dimensional data into much simpler format by projecting the data into low-dimensional representation. And many researchers are using a linear approach, such as principal component analysis, by identifying principal axes that are not correlated to each other and have maximum variance. Indeed, the PCA is very simple to implement, but many real-world data sets have nonlinear characteristics, which a PCA mapping fails to encapsulate. So many recent studies adopt nonlinear techniques, such as diffusion map embedding, to reduce the dimension by reorganizing the data according to its underlying geometry. In this low-dimensional space, the data with similar patterns are located closely, while those with different patterns are located far apart. And I want to introduce these kinds of dimensional reduction techniques that can be applied for our neuroimaging connectome data. The main idea is to reduce our connectome data into a series of continuous dimensions, which represent the principal axis of the whole brain connectivity pattern. And I will refer this low-dimensional feature as a connectome manifold or eigenvector, and we usually call this as gradient. The gradient is a vector field that has gradually changing patterns with a specific direction. And someone may confuse this concept with mathematical derivatives, but it does not represent that concept. It just means the gradually changing spatial patterns. And you can easily find this concept in neuroscience. The retinotopy represents the gradually changing visual fields, which is very useful for defining different visual areas from early visual to high order visual cortex. And tonotopy is quite similar to the retinotopy, but it is defined in the auditory cortex. And this gradual pattern is defined based on the different tones of the auditory signal. And here, the main methodology is this one, the connectome manifold. So let's assume that we have connectome data. It can be either functional or structural connectivity. And we can estimate the low dimensional manifolds by applying some kinds of dimensionality reduction algorithms. And this dimensionality reduced feature is the connectome manifold. So the concept is very simple. And the toolbox to construct the manifold is openly accessible in our website, built by Reindel and Walid in our lab. And it is very useful for constructing the gradients, as well as aligning the gradients between different subjects or different imaging modalities. And I brought this example from Daniel Maguglio's paper to tell you why this approach is promising. And let's say the first gradient of the functional connectome data as an example. So if you can see the panel A, you can see a very interesting pattern. The low-level sensory areas have the strong negative values, while the high-level transmotor areas have the strong positive values. And this indicates the first principal axis represents the spatial hierarchy of the brain. And the similar patterns were observed in the Macroscale data as shown in panel B. And the differences between the gradients are expressed in panel C. The first gradient shows the axis from the lower level idiotypic to the higher level parambic areas, while the second gradient extended from the visual to auditory areas. So we can say that the first two gradients have different cortical representations. So we can summarize the connectome manifolds. The gradients are the low dimensional representations of the connectivity patterns, which can be applied to our multimodal neuroimaging data. And each manifold provides different cortical representation, which is very useful for understanding the connectivity patterns. And indeed, these techniques have gained a lot of traction to study large-scale principles of microstructure neuroimaging data, as well as functional data in neonatal and autism brain. And also, these techniques can be applied to a specific brain region, not to the whole brain, such as hippocampus and insula. And these gradient techniques are not only performed by our lab, but also widely used in many other promising studies. For example, identifying gradual patterns of the specific brain regions, such as sensory-motor cortex, subcortical regions, and cerebellum. And also, it is used for identifying the location of the brain lesion in the manifold space. However, this approach remains underexplored in the study of structure connectivity based on diffusion-weighted imaging. So in this talk, I will introduce how the structure connectome manifolds can be applied to our studies with 3 different topics. The studies include the first, the correspondence between brain structure and functional dynamics. Second, the maturations in structure connectome during adolescence. And lastly, the connectome perturbations in the atypical neurodevelopmental conditions such as autism. So let's start from the first study, the structure-function coupling. In this study, we tried to construct the framework for linking the brain structure and functional dynamics using diffusion and functional MRI that obtained from the HCP database. The main methodologies are the manifold learning approach for quantifying the structure connectome data and the hidden Markov model to represent the functional dynamics. And then I will first briefly introduce the background of this study. In neuroscience, there is a core assumption that the brain structure governs ongoing function, which means the brain function could be established according to the structure patterns of the brain. And many studies try to understand the underlying mechanisms of the correspondence between brain structure and function in terms of brain connectivity. The structure connectivity represents the anatomical connections between the brain regions, and it can be inferred via diffusion MRI by approximating structure wiring in white matter through the tractographic algorithms. And functional connectivity is the statistical relationships which can be calculated by the correlation of the time series between different brain regions. And here, the important question is how the brain structure and function are related in terms of a large-scale brain network. In a previous macaque study, they found very similar patterns between a seed-based functional connectivity and the structure fiber tracks. Here, if you set a posterior cingulate cortex as a seed, the functional and the structural connectivity show very consistent connectivity patterns through the default bone network. And this result represents that there could be the correspondence between the structure and functional connectome, at least in this specific brain network. And many other human studies try to associate the brain structure and function using the large-scale connectivity information. For example, by estimating joint singular vectors from the structure and functional connectome data, or simulating the functional connectivity based on the structure connectome information. However, there is one thing we have to consider. Most of the structure-function coupling studies were performed based on the static functional connectivity. The brain, however, is an inherently dynamic system where function and cognition involve the time-varying network reconfiguration patterns. Indeed, in both humans and non-human primates, the links between the brain structure and specific cognitive function have been well established in a static manner. Although these studies highlight the links between the specific neural patterns and the particular aspects of the cognition, such stationary analyses are not well suited to understanding how the brain flexibly changes between different modes of operation. So now it's time to focus on the correspondence between brain structure and function in terms of the dynamic functional connectivity. Here, what we want to explore is how to integrate the brain structure and functional dynamics. So I will introduce a framework for combining the brain structure connectome with the functional dynamic patterns. The brain structure was quantified using the structure connectome data that derived from the diffusion MRI. And after constructing the structure connectivity matrix, we applied the manifold learning techniques to estimate the principal eigenvectors. And we selected the two components that showed enough variance to explain the total connectome data. And I will refer these two manifolds as structure gradients 1 and 2. So as you can see the figure in the bottom, the first gradient differentiated the sensorimotor cortex from a medial prefrontal ankle, and the second gradient extended from the ventral to dorsal areas. And then the functional dynamics were quantified using the hidden Markov model. First, we concatenated the time series of the whole subject and then applied the hidden Markov model. It automatically estimated 14 brain states, but it's quite complex to analyze all these 14 different brain states. So we try to simplify these brain states. We apply the modularity detection algorithms to the transition probability matrix, and we could find the two distinct clusters. And I will refer these clusters as the metastates. So we can say that these two brain states have different transition patterns to each other. And these metastates show very interesting patterns. The first metastate seems like it has high activity in the sensorimotor areas, and the second metastate has strong activity in the default mode and frontoparietal networks. So we can say that the first metastate can be depicted as a low-level sensorimotor state, and the second metastate as the higher-order transmotor state, which indicates the temporal spatial hierarchy of the brain. So now we are going to associate the brain structure and functional dynamics. Here we define the two low-dimensional manifolds from the structure connectome data, and also we have the two activity maps from each metastate. And we will associate these structure gradients with the transitions of the metastates. As we have the two metastates, we can quantify 4 different combinations of the transitions within each metastate and between the metastates. And we simply calculated the Pearson's correlation between the structure gradients and the differences in activity patterns between the metastates. And we could find a strong relationship between the first structure gradients and all the transitions except for within the metastate 2. And when you combine the first and the second gradients based on the linear regression, the magnitude of the association was slightly improved. So we can say that the brain structure can explain the lower level transitions, but it is not well suited to explaining the higher order transitions. Then the remaining question is why we couldn't explain the higher order transitions using the structure connectivity. To explore this, we adopted several connectome topology measures, especially rich graph taxonomy, connectivity distance, and network communications. We hypothesized that there must be different anatomical communication mechanisms that differentially affect functional dynamic transitions. Because of the limited time, I will skip the details of each method, and I will just let you know what we found in brief. The primary finding is that we could find the obvious differences in the connecting topology measures between the metastates. The low-level sensory-motor state follows the communicative principles of network diffusion, and it usually occurs in the local nodes with the short-range connections. And in contrast, the high-level transmotor state occurs more along the shortest path, and it processes parallel information between the heavily distributed transmotor hubs. So taken together, we found that the structural connector manifolds can explain the dynamic transitions that involve the low-level sensory motor states, but the magnitude of the structure function coupling becomes weaker if the transition is anchored to the higher-order transmotor state. And we also found that these two dynamic states follow different communication mechanisms of the structural network. Okay, then let's move on to the second study. The main idea of this study is to see how does macroscale structure connectome organization changes during the typical development using a longitudinal data set. Among the developmental stages, adolescence is a time of profound changes in whole brain network organization. Indeed, the early cross-sectional and longitudinal MRI studies observed morphological changes in gray matter volume, with initial gray matter increases until the late childhood and followed by a decline for the rest of the lifespan. Also, in the typical developing condition, the degree of cortical thinning is apparently observed in the prefrontal cortex during adolescence. And it has been shown that this cortical thinning is highly associated with the development of social intelligence, indicating that the adolescence development is important for overall maturations in cognitive functions. In addition to the gray matter volume and cortical thickness, it has been shown that the myelinated fiber density showed an increasing trend across age. The more recent work explored the changes in intracortical microstructure, capitalizing on myelin-sensitive contrast, such as magnetization transfer ratio, which suggests the overall increases in adolescence, and together with the depth-dependent shifts in the intracortical myelin profiles. And several studies suggested the connectome reconfigurations in adolescence based on the resting state fMRI. And these studies showed that during development, the brain organization is remodeled from a more local towards a more distributed architecture through the relative strengthening of the long-range functional connections. So collectively, these findings suggest the measurable trajectories of cortical morphology, microstructure, and functional connectome organization. But still, we lack an evidence of how structural connectomes are remodeled during the brain development. So what we want to explore in this study is to assess the longitudinal reconfiguration patterns of the structure connectome data that derive from the diffusion MRI. So in this study, we took an advantage from a large-scale accelerated longitudinal data set that obtained from the Neuroscience and Psychiatry network cohorts. And similar to the previous study, we first generated the structure connector manifolds using the diffusion MRI. And here, we selected 3 structure manifolds. The interesting point here is the manifold eccentricity, which is defined as the Euclidean distance between the center of the template manifold and all the data points in the manifold space. And through this approach, we can simplify the multivariate manifold representations into a single scalar, which represent expansion and the contraction of the eigenvector in the newly defined manifold space. So to see the longitudinal changes of the manifold eccentricity across age, we constructed a linear mixed effect model. The random effect of the subject was added, and the model was controlled for sex, size, and head motion. Interestingly, we could find a significant effect in the higher-order cognitive functioning areas, especially in the prefrontal, temporal, and parietal cortices. As you can see at the scatter plot on the bottom, we can see the manifold was expanded in these areas, which means these higher order transformer areas have a trend of expansion of structure manifold as an individual getting age during adolescence. So now we wanted to see whether this manifold organization is related to the conventional graph theoretical measures. And we could find a strong relationship between the changes in manifold eccentricity and those in the within module degree, indicating the expansion of the manifold eccentricity is related to the integrated topology within the transmotor areas in the graph. And the significant association was also found with delta degree centrality, which is very consistent with the module-related measures. And in addition, we calculated the connectivity distance and correlated with the manifold eccentricity. And they also showed the significant relationships, indicating the manifold eccentricity expansion is also related to the physical connections between the brain regions. And okay, then it has been shown that some brain structures of cortical thickness and myelin content significantly change across the age. So we replicated the prior findings using our own data and found very consistent results. The cortical thickness and the skewness of the magnetization transfer, which is the myelin sensitive index through the cortical layers, showed decreasing trend according to the age in a longitudinal setting. And to see whether the manifold changes are affected by this cortical morphology and microstructure, we estimated the mixed model effects after controlling for the cortical thickness and myelin content from the structure manifolds. And notably, we could see almost unchanged results, which means the structure manifold change across age independent to the brain structures. And we additionally performed the developmental enrichment analysis to contextualize the longitudinal manifold changes with respect to the patterns of the postmortem gene expressions. And this analysis highlighted marked associations with the expression of genes that enriched from childhood to adulthood in the cortex, thalamus, and cerebellum. Then we wanted to see how the structural connectivity of the subcortical areas change according to age. So we assessed the longitudinal changes in streamline strength between the subcortical regions and the identified cortical areas. And we found that the stratum, including caudate, foramen, and pallidum, showed significant strengthening of streamlines connecting cortical areas. And notably, the caudate showed the largest changes in the streamline strength. And finally, to assess how the structural connectome changes are related to the development of cognitive intelligence, we utilize supervised machine learning to predict full IQ at follow-up using structural connectome information of the identified cortical and subcortical regions. And we found that these structural connectome features were quite useful for predicting IQ, especially the regions showing strongest predictive validity for IQ were prefrontal, parietal, and temporal cortex, as well as the caudate. And when we decoded the probability map of the brain regions using Neuroscience, we found that these areas are quite a lot associated with the higher order cognitive and social types. So in summary, our work provides a novel perspective how the structure connected manifolds mature during adolescence that can bridge the molecular processes and cognitive behavior outcomes. And the final study I want to introduce is a multiscale study using the autism data. The autism is one of the most common neurodevelopmental conditions showing impairments in social interactions, language abnormalities, and repetitive behaviors. And it is quite an appealing topic to explore because it is a very common disorder, and also it has quite heterogeneous characteristics of the individuals in terms of behavior symptoms and etiological factors. In the previous functional neuroimaging study, Martin et al. Found dominant hypoconnectivity, particularly for cortico-subcortical and interhemispheric functional connectivity. In contrast, another study showed increased functional connectivity between the primary sensory networks and subcortical areas, especially in thalamus and basal ganglia. In addition to the macroscale neuroimaging studies, more synoptic-level studies found that the autism patients show imbalances in microcircuit functions. Especially, autism patients show sensory deficits compared to the typical developments, specifically the altered link between visual suppression and GABA levels, and atypical size of the receptive fields in the visual cortex. And in addition to the primary sensory cortex, excitation and inhibition imbalance was observed in the association cortex and limbic system, which indicates the autism is related to the low-level sensory deficits as well as atypical higher-order cognitive functioning. So we can say that the autism spectrum condition impacts multiple scales of the brain organization, from macroscale anomalies as well as the local changes in microcircuit functions such as excitation, inhibition, imbalance. However, we currently lack an overarching framework that can bridge the topological changes at macroscale and microscale dysfunctions in autism pathophysiology. So the aim of this study is to examine how abnormal macroscale organization relates to the microcircuit imbalances and furthermore, the altered gene expressions in autism. Similar to the previous study, we estimated the low-dimensional manifolds using the structural connectome data. And here, we selected the 3 manifolds that explained almost 50% connectome variance. And using the multivariate analysis, we compared the 3 manifolds between the controls and autism patients, controlling for age, sex, and sight. And we observed the macroscale distortions in autism patients in multiple networks with primary effects in the sensory and somatomotor areas, as well as the association cortex. And stratifying the effects according to the laminar differentiation of the cortical hierarchies, we identified the peak effects in the idiotypic areas. And similarly, when you analyze the effects with respect to the 7 intrinsic functional communities, we also observed the strongest between the differences in the somatomotor network and followed by the higher order, the fourth mode network. And these results are, again indicate that the autism patients have low-level sensory deficits as well as atypical high-order cognitive functioning. Okay, then in addition to the macroscale findings, we implemented a large-scale biophysical dynamic circuit modeling to quantify the microscale brain organization. In comparison to other models that also include the synapse-level parameters, this model has more synoptic scale, allowing for the structural function simulations with modest parameter complexity. And this model inversion approach can successfully estimate the microcircuit parameters, especially the recurrent connection and the external subcortical input. To assess the relationship between the macroscale connectome information and the microcircuit neurodynamics, we correlated these regional changes in microcircuit patterns and the macroscale anomalies. And we observed a significant correlation between the overall degree of manifold distortion and the increases in the excitatory subcortical inputs. And to provide the additional neurobiological context of our macroscale findings, we performed the transcriptomic association analysis. Here initially, we correlated the t-statistic map of the macroscale connectome distortions and the postmortem gene expression maps provided by Allen Brain Atlas. And the gene symbols that passed for the significance level were fed into the enrichment analysis, which involved the comparison against the developmental expression profiles. And this analysis highlighted the associations between the multivariate patterns and the genes that are strongly expressed in the early childhood and adolescence, especially in the thalamic and cortical areas. It is quite interesting because in the microcircuit analysis, we found significant interactions between the cortical manifolds and the subcortical inputs. So combining that finding with this current enrichment results, we can infer that the autism can be characterized as a multiscale disorder that point to the dysfunction in the thalamocortical interactions. And finally, we added a biological meaning to our macroscale findings by associating the behavior symptoms of autism. And we employed the elastic regression framework to predict the symptom severity scores measured by ADOS. And similar to the macroscale findings, the structure manifold features in heteromodal association areas are particularly useful in predicting impairments in low-level repeated behavior symptoms, as well as the high-order social and cognitive deficits. But they were not very sensitive in predicting atypical communication abilities. And this inability remains to be investigated further, but it may potentially relate to other aspects of the autism heterogeneity, including speech onset delay that affects only a subgroup of the autism individuals. Okay, then here in this study, we suggested an overarching framework that consolidates macroscale connectome to the microcircuit parameters and the gene expressions. And I believe this approach would be a good framework for conducting future multiscale studies. If you want more details on the multiscale and neurodevelopmental studies, please visit bioRxiv to read these preprints. Okay, then let's conclude this talk. Today, I introduced a broad concept of connectome manifolds and especially emphasize how the structural gradients derived from DWI can be applied to the typical and atypical developmental conditions. And they were actually useful for providing important models for brain function and for tracking brain maturations and predicting cognitive intelligence, and also for better understanding the brain disorders associated with atypical development. So thank you for listening to my talk, and thanks to all my collaborators, especially Boris and Mika lab members, and also to funding sources. And especially, thanks for having me today to this wonderful seminar. Thank you.
Thank you for the talk, Bo-yong. So now the floor is open for questions. So you can either type the question in the chat box, or you can turn on your video and unmute yourself and ask the question to Bo-yong directly. So there is one question in the chat from Nathan. I'll read it to you. Very interesting talk. Can you clarify how your measure of manifold eccentricity relates to dispersion of the manifold as characterized by Bethlehem et al. In bioRxiv? And how do you interpret what manifold eccentricity means in terms of brain organization and development?
Thank you for the great question, Nathan. I'm not exactly familiar with the dispersion, but I think, as I know that the manifold eccentricity, the differences between manifold eccentricity and the dispersion is just between, is it? So the manifold eccentricity is calculated between the center of the template manifold and all the data points. And the dispersion could be calculated between the different regions. So that can be the differences between the eccentricity and dispersion, as I know. And the major advantage for calculating the manifold eccentricity could be we can quantify how much the manifold is expanded or contracted in a very easy way. So it could be the new method for quantifying how the brain organization is quantified. So yeah.
It could be some type of range of connectivity or some granularity of connectivity profiles that are represented, and maybe then has some implications for functional dynamics, possibly.
Yeah.
But yeah, I also think that the difference between the dispersion and the eccentricity is correct. That one of them is a global feature and the other one is between the different points. So there's another question from Sophie. Great talk. How do you think your patterns relate to more evolutionary mechanisms underlying brain development, such as expansion, so cortical expansion? And do you think that these manifolds can change, I assume, throughout evolution?
very interesting question. And I'm not exactly sure, but I'm sorry that can you explain can you read the question again?
So basically whether the patterns that you observe also relate to evolutionary mechanisms. So in other words, whether you would expect these manifolds to change throughout evolutions when you compare, for example, non-human primates and humans.
Oh, can I also speak, or is this?
Yeah, sure.
Bad? Okay, yeah, I just wondered, like, you showed these manifold changes in autism, but I wondered, so it was actually two questions, and so I wondered, like, with changing environmental demands, can you expect, for example, in autism that, for example, training or like an intervention could change also the manifold disruptions again that you observe, or is it some static difference?
I'm sorry that I didn't exactly understand your question, but
So yeah, to clarify, you find differences between autism spectrum disorder and healthy controls, right?
Yeah.
And these relates to also gene enrichment patterns and like at many levels. But on the other hand, there's of course also hope to have like therapy or an intervention that might alleviate some of the challenges that people with autism face. So I wondered if you think that these manifold differences between healthy controls and people with autism spectrum disorder might also be less different with certain interventions.
Yes, that makes sense, but that should be investigated further. But I think it makes sense. So maybe for the intervention stuff, then yeah, sure, the manifold will be changed, but
Sophie, what's your perspective on this? Because you have done some training studies.
Interesting question. I think in some level, the brain is adaptive and we live in an environment in which we constantly need to adapt. So it can, ideally, I'm now writing up a study where I show that there can be changes that also relate to brain dynamics. But on the other hand, I don't know if in autism, these changes are so easy as in healthy controls, right? Because it's a challenge to change a normal person. Like I want to change all the time and I am still myself. But I guess with people with a disorder, it's even more like the scope in which they can be flexibly adaptive to environment, or that you can help them with therapies is maybe more narrow. So maybe their system is not so changeable in certain directions. But I do think such measures as these manifold and more holistic approaches to brain disruptions might help us also to guide what is a fruitful intervention, for example. But I guess there's a lot to learn still.
Yeah, I got your point. So maybe I think in my point of view, the structural connecting manifold will not change a lot, I think, but the functional manifold could change due to the intervention, because the manifold sometimes reflects the functional connectivity between the different brain regions. So the manifold could change according to the intervention, I think. Even they are the normal population, the health controls.
Yeah, thanks so much. I really like your work, so good job.
Thanks. Thank you.
It's possibly also depending on which regions you want to target. So maybe the regions that have a lot of synaptic plasticity, maybe in those features are more, you have like more ability to adapt with interventions. So higher order areas, maybe training can result in effects that are easier to detect. Maybe lower level areas that are relatively stable, it's not so easy.
Yeah, but there are, I guess, some juggling studies that focus actually more sensory mechanisms, right? But yeah, I guess it is, I can imagine there's also a differential effect of to what extent an intervention can change your brain or the system, and also that it also is not unique, like not the same in disorder in all regions, right? So maybe it's easier or less easy in unimodal regions relative to transmodal regions in autism, relative to controls, right? I think that there's a region or network specific element to it as well. So there's no global explanation, I can imagine. But yeah, I'm also in a different time zone, so I'm also a bit tired. But yeah, I really think the work is super cool. So
Thank you, Sophie.
Thanks, Sophie. So if there are no further questions, we can close the session. Thank you all for joining. And if you have further questions, you can reach out to Bo-yong and follow him on Twitter.
Thank you.